many of you know, my research focuses on biomarkers of aging, many different types of biomarkers. But today I will talk about epigenetic clocks. I will highlight applications to human clinical trials, but also preclinical work. All of our biomarkers are based on cytosine methylation, which is an epigenetic marker. And the first generation of epigenetic clocks was focused on estimating chronological age or calendar age. And one of the insights was that these pan-tissue biomarkers apply to all sources of DNA, all tissues, all cell types. And when you can estimate methylation age, or also known as epigenetic age, you can form a regression line, which in essence is the expected value for each person. And when people lie above this expected value, they are known to exhibit positive epigenetic age acceleration. It's just the terminology. So if you're above your expected value based on your chronological age, you are arguably older than expected. And research has shown that positive epigenetic age acceleration is associated with higher risk of mortality and other and morbidity. But we are now at the stage of developing the second generation epigenetic clocks. Well-known examples are DNA methylation PhenoAge and GrimAge. And these clocks should be interpreted as estimators of mortality risk. They are not estimators of chronological age. And our most accurate predictor of lifespan based on blood is known as GrimAge. And so if you are in the top 5% of fastest agers, according to the GrimAge clock, you face a mortality risk that is 2 times that of the average person of your age group and your smoking status group. I briefly want to mention GrimAge predicts lifespan even if you never smoked in your life, or conversely, if you are in a group of heavy smokers, it will still predict lifespan. So any stratum formed by smoking status, age group, and sex, and of course race and ethnicity, any of these groups could benefit from the GrimAge clock as when it comes to mortality prediction. I just wanted to show you here one meta-analysis of the GrimAge clock. Here, the key thing is perhaps the p-value, 10 to the minus 75. We analyzed the Women's Health Initiative, Jackson Heart Study, Inchianti, Framingham Heart Study. And so the p-value is very significant. The hazard ratio seems small, except that it's associated with 1 year of age acceleration, you know. And so if you have 8 years of age acceleration, you need to raise the hazard ratio to a power of 8. So it adds up pretty quickly. We are now in the process of trying to qualify epigenetic biomarkers such as GrimAge as surrogate endpoints for human anti-aging studies. And this is a very hard hurdle to take. And I just mentioned a couple of bullet points. First of all, you need to show that a biomarker has strong cross-sectional associations with mortality and other outcomes. And there are over a dozen papers by independent groups that validate the original claims about GrimAge. People from all over the world, from Europe, from New Zealand, and so on, have validated that GrimAge predicts lifespan in cross-sectional studies. There's no debate anymore. The second step for GrimAge are really longitudinal studies, which is a different cup of tea. And we need to carry out longitudinal studies in order to establish causal relationships, because we want to show that if you have an anti-aging treatment that reverses GrimAge, that you actually end up prolonging lifespan. You know, that's a very strong claim, and which is hard to establish, but longitudinal studies help in that regard. So to achieve that, we want to show that the rate of change in GrimAge is associated with increased hazard ratio, right? Imagine you have 2 blood draws or 3 blood draws. You can measure the rate of change of GrimAge in a person, and you want to say, because it shoots up, this GrimAge shoots up, therefore this person is in trouble. And we have some interesting data in this regard, which I will show to you in a second. Another way of establishing causal links is to use genetic studies. That's actually one of the main purposes of genetics, to help you with causal statements. And we just put a large-scale GWAS into bioRxiv, where we have shown that polygenic risk scores for GrimAge are strongly associated with parental longevity. And I'll talk about it in a second. But then bullet point 4 is known as analytical validation of a biomarker. It's also known as test-retest variability. And again, you want low test-retest variability. Now, I mention all of these points as a high-level summary because these bullet points have been established, and I will now walk through the data. So regarding this claim that the rate of change in GrimAge is associated with increased hazard of death, Alexandra Binder has shown that in the normative aging study, this rate of change, which was measured using a linear mixed model, was associated with higher mortality. I did mention these polygenic risk score analyses. So Daniel McCartney and Riccardo Marioni led a study where they analyzed methylation data and SNP data from 41,000 individuals. And this very large sample size allowed us to identify 137 genome-wide significant loci for several epigenetic biomarkers, my original pan-tissue clock, the GrimAge clock, the PhenoAge clock, and so on. But importantly, this study for the first time presents polygenic risk scores. So if you have SNP data, you can now check whether a polygenic risk score for GrimAge is associated with mortality. And I want to mention all of these data are publicly available, so I invite you to check it. What I can tell you is we do find very strong, and strong is the wrong word, significant, statistically very significant associations between longevity and mortality and a couple of other exposures. Now, as I mentioned, biomarkers will only be useful if they have low test-retest variability. In other words, if you go to the doctor and you measure your GrimAge on Monday, and then you come to the doctor's office a week later, and you again measure it, that measurement better be very similar. Why? Chances are you didn't age in 1 week, you know. The ICC values above 0.90 are excellent, and GrimAge reaches these, you know. So, and here I show you some results. We have generated data with technical replicates, many different data sets. Here I present you findings from 5 different data sets. And you see, these are ICC values. You notice they are very close to 1. I mean, this means GrimAge has exceptionally high test-retest accuracy. The one value where it's low, it's when you compare buccal swabs, you know. But so why is it low? Well, because GrimAge is really a biomarker for blood, you know. So it's not expected to work in buccal cells. We like GrimAge because it's our best epigenetic biomarker for time to death, time to coronary heart disease, time to cancer. It relates to early age of menopause. I mentioned that an ingredient of that biomarker is an estimator of smoking pack years.
Nice.
And interestingly, our methylation-based estimator of smoking pack years actually outperforms self-reported smoking pack years in 3 different studies. So it's interesting. I want to draw your attention to the fact that epigenetic clocks are already being used in human clinical trials. And here I mention this study from Greg Fahy and Bobby Brooke, where Greg Fahy applied it to a very interesting treatment where he regenerated the thymus. I'm sure Greg will talk about it. And I just wanted to mention that a very interesting side effect of Greg's treatment was that it actually rejuvenated the age of blood samples of these people in this phase 1 clinical trial. And here I show you the figure from his paper. So the x-axis shows you the months from trial onset. So we had 2 blood draws at baseline, and then we had follow-up blood draws. And each panel corresponds to a different epigenetic clock. And in panel D, I show you how GrimAge goes down. So after 12 months of this treatment, there was an age reversal of 2.5 years, according to the GrimAge clock. So that was quite an exciting finding. The limitation was that this was a phase 1 clinical trial, but I'm very happy that Bobby Brooke and Greg Fahy are now initiating the so-called TRIIM-X expanded pilot clinical study, and they're enrolling people, estimated enrollment 85 people, where they will revisit these analyses. So if we're lucky, in a year or so, we will know whether this thymus regeneration procedure validates and rejuvenates the immune system. I want to now come to a different topic, preclinical studies, more generally epigenetic aging studies for mammals. And you want, in the following, we need to distinguish age from maximum lifespan. So, as you know, a central question of biology has been, why do similar species, such as mammals, have markedly different maximum lifespans? There's a vast body of literature that goes back to Aristotle, but beautiful articles from the research community which have looked at that question, especially when it comes to ecological parameters. And all of you know that animals who are bigger or who can fly, or more generally, animals that can evade predators live longer. But I, of course, look at it through the lens of methylation, and we have found that CpG methylation correlates strongly with maximum lifespan across species. So here I show you results for a single cytosine, one cytosine. And notice the correlation is minus 0.64 for log maximum lifespan. Each dot is a species, okay? Now, more generally, you can do so-called EVOS studies, epigenome-wide association studies. And here we have both generic regression analyses and also phylogenetic regression analyses. Just wanted to show you our data. The y-axis is log, is the logarithm of the p-values. We see p-values 10 to the minus 25 or so, but we really find hundreds, if not thousands, of cytosines that actually correlate very significantly with lifespan. And that holds even after you adjust for phylogeny. And so stay tuned. There's some papers coming out where we describe some of our findings. I want to now move to the question of measuring chronological age, not maximum lifespan. And I start out with the finding for elephants. You may remember there are two different species of elephants, the African elephant and the Asian elephant. And we had blood samples, and if you combine these two species, you can develop a very accurate clock for blood from elephants, correlation 0.96. By the way, when I show you results in the following, it's all based on a cross-validation analysis. So these are not training data. These are, in this case, leave one out cross-validation. And so the very high correlation. Now, if you focus on a single species in panel B, this is African elephant, panel C, Asian elephant, very accurate. But look at panel D, there's something crazy going on, because here we combined elephant data with human data, okay? So what we have here is an epigenetic clock that applies to three species at the same time: human, elephant, Asian elephant, and African elephant. And this clock is basically a single formula. It's one regression model. You give us a blood sample from any of these species, we can measure age with a correlation 0.97. Very accurate. So now let's move to another species, cats. I remember that we adopted a cat and we never knew its age, but nowadays this would be an easy problem for me because we have a very accurate clock for blood samples from cats, correlation 0.94. And for cats, we did the same exercise. We said, can we build a clock that applies both to cats and its owner, And that's in panel B. So we have these human cat clocks. And panel C shows you how the human cat clock performs if we only analyze blood samples from cats. We still get a correlation 0.97. So if you forgot when you were born, or if you don't, and you don't know your cat, you can get your blood drawn at the same time, and we measure your age. Same for dogs. I mean, we have very accurate epigenetic clocks for different dog breeds. This is a collaboration with Elena Ostrander, Andrew Hogan, and others, where we analyzed blood samples from 51 pure dog breeds, and again, correlation 0.96. What about the naked mole rat? A very interesting animal to all aging researchers, an animal that does not age on a phenotypic level. But interestingly, when you do look at methylation, you can build an epigenetic clock for that animal. And that's a seemingly paradoxical finding, because how is it possible that this molecular marker of aging picks up that this animal ages, even though on a phenotypic level it does not? And there are really several interpretations. I let you think about it. But one interpretation is that the naked mole rat developed mechanisms to deal with that noise, so it effectively can deal with these methylation changes. What about rats? We have, again, many rat clocks, actually. So we have a pan-tissue clock. Here we analyzed, I want to say, 8 different tissues from rats. And we have a pan-tissue clock. We have a clock for brain samples from rats, a clock for blood samples from rats. I didn't mention we have a human rat clock. So again, very accurate clock that applies to both species. In a couple of days, we will put a paper in bioRxiv about epigenetic studies in bat species. So Jerry Wilkinson spearheaded a study where we looked at 26 different bat species of very different ages. You may know some bats live for over 40 years, but other bat species live short lives, maybe 5 years. And we have developed very accurate clocks for these different bat species. You can give us a wing punch from any bat. We can estimate its age, And but also we have cytosines that relate to lifespan in bats. So I invite you to learn the biology from these papers that will be coming out. Now, we are, of course, very interested in building what we call universal clocks. So let's first look at the right panel. Here I show you different species. The species are labeled by phylogenetic order. So phylogenetic order 1 are primates. Then I let you look through all of them. But we have here many different species. In total, we collected data from 160 mammalian species. And the question is, can you build a so-called universal mammalian clock that applies to all of these mammalian species? And here I show you results. Again, these are unbiased results. The emphasis, these are test data. And so in a test set, correlations are very high, 0.98. But panel F is perhaps more interesting for people who have a species that is not part of our training set. Because again, we have 160 species, but obviously there are hundreds of additional mammalian species. So how accurate would be this universal clock if you give us a completely novel mammal? And the answer is answered in panel F, because this reports so-called leave one species out cross-validation. But even here, you see a remarkably accurate clock correlation. The median across species is 0.88, so almost 0.9 when it comes to correlating age with our universal clock estimate. So in conclusion, I think I've talked about human clocks, and what I didn't highlight is the human clocks differ actually greatly in terms of predictive accuracy for mortality. The GrimAge clock would be much better for predicting mortality than my original pan-tissue clock. I also showed you that methylation actually strongly relates to differences in maximum lifespan across species. Coming back to humans, I want to emphasize that methylation is predictive of mortality even after adjusting for many risk factors. Standard clinical biomarkers could be put into a regression model, and GrimAge would still be very significant. So methylation adds an additional layer to standard clinical biomarkers. I also want to emphasize that methylation clocks will not replace standard clinical biomarkers. Any anti-aging trial will have to measure standard biomarkers such as glucose level, blood pressure, and so on. There's no way around it. When you study and design your own studies, you need to think very carefully about what cell type you want to profile. Do you want to look at blood? Some anti-aging interventions have only a detectable effect in buccal swabs. For example, a menopausal hormone therapy seems to affect buccal methylation age, but not blood. And I'll stop by acknowledging the many people who have helped in my own lab, Akelu, and my many collaborators, Ken Raj, Bobby Brooke, Greg Fahy, and so on. Thank you very much.